Hello, everyone. Thank you for coming to this pre-ESRS webinar interview with keynote speakers. My name is Melissa Derry, and I'm an assistant professor from Lehman College in New York, and a member of the SRS-TT. The SRS think tank, or SRS-TT, is a group of early career researchers in the radiopharmaceutical sciences within the SRS, which aims to build up content and programming focused on the needs of early career researchers. We currently have 13 members, and our goals include providing opportunities for networking and access to experienced researchers and their expertise, as well as career planning and training. I'd like to introduce two other members of the SRS-TT, Valerie Redchenko from Triumph and Emma Carbo from Simon Fraser University. A new initiative of the SRS-TT is to start a webinar series focused on careers in radiopharmaceutical sciences to help give context to trainees as they make their choices to shape their own career development and paths. As a complement to all the great scientific talks coming up in the ESRS meeting, we decided to do our very first webinar focusing on career profiles of our highly esteemed plenary speakers. In planning this event, we wanted to think beyond education and job training and get into the personal journeys of people in our field. Without further ado, I'd like to introduce our two guests of honor who were kind enough to join us for today, Professor Nicole Pamme and Dr. Robert Mach. Nicole Pamme works in the chair in the analytical chemistry at the University of Hull in the UK, in the Department of Chemistry and Biochemistry. She's a fellow of the Royal Society of Chemistry and a fellow of the Higher Education Academy. She currently also serves as the president of the Chemical and Biological Microsystems Society. Her research revolves around the study of microfluidics lab-on-a-chip devices for on-site environmental analysis, and point-of-care clinical diagnostics, as well as synthesis and quality control of radiopharmaceuticals. You can hear more about her work during her plenary presentation, Compact Cyclotrons and Lab-on-a-Chip, a pathway to dose-on-demand PET tracers, on Wednesday the 19th. Robert Mach is a Britton Chance Professor of Radiology and Director of the PET Radiochemistry Program at the University of Pennsylvania in the US. He is a past president of the Radiopharmaceutical Sciences Council and the Society of Nuclear Medicine and Molecular Imaging, and a former board member of Board of Directors of the Society of Radiopharmaceutical Sciences. His current research projects include the development of radiotracers for imaging addiction, Parkinson's disease, cancer, inflammation, and more, as well as studying fundamental cancer cell biology. You can hear more about his work during the plenary presentation, Radiochemistry and Total Body PET, on Monday the 17th. Lastly, I'd like to turn things over to Valerie, who will host the actual interview. Thank you.
Yeah, thank you very much, Melissa. And thanks a lot for finding the time and join us for the interview today. And we're hoping that we can learn more from you about how you develop this career and how you get to this great stage where you are right now. So maybe I can start then with the first question, which is just to, can you give us a brief summary on your career path, how you get to the stage where you are and how you develop your career. And I don't know what the order, Nicole. Do you want to start first and then Robert?
Yeah, it's the ladies first thing, right? That's the crux. Yeah. That it's me here. It's okay. Yeah. So thank you very much for having me. It's the first time I'm doing a seminar like this. So I'm not a traditional radiochemist. I think I started working with radiochemistry only in 2008 when I was already a senior lecturer or a lecturer at a university. So I grew up in Germany, as you can probably hear from my accent that's still around. I come from a relatively rural area in Germany, first generation of children that would do A-levels and go on to university to study. So I liked a lot of things at school. I I was relatively good in relatively many subjects, but somehow I had a bit stronger affinity to chemistry than some of the other options I was considering. Probably a lot teacher influenced, I guess, at that time. And yeah, I went to study at the University of Marburg, which is sort of in the middle of Western Germany, a relatively old university, small, quaint university town. And 1 year during that time, within Europe, we have this Erasmus exchange program. So I had a chance to go to the UK. I went to Imperial College London for about half a year, and then worked in a company near London for another half year. So I spent about a year in London before I returned, finishing my chemistry degree. And I started specializing a bit towards the end of the degree in analytical chemistry. So I knew I wanted to do a PhD thesis in that area. And to be honest, in Germany, you don't even, when you study chemistry, the PhD is just what everybody does. So once you start studying, you kind of know that you're going to be doing a PhD afterwards. So it wasn't so much a question of if you're doing a PhD, but rather more in what topic and where specifically. So I applied at a few different places, but funnily enough, I ended up at Imperial College again in London with a professor called Andreas Manz, who was one of the pioneers in this area of lab-on-a-chip and microfluidics. I never met him when I was there earlier because I was doing inorganic chemistry at that time. But yeah, so I spent about 4 years at Imperial College studying for the PhD, and I was also a teaching assistant at the time. And yeah, then I had an opportunity to go to Japan. So I went quite far from the rural Germany and worked in a materials research institute near Tokyo, not directly in Tokyo, but quite close to Tokyo for about 2 years. And yeah, then I applied for the lecturer position at the University of Hull, which is sort of on the east coast in the United Kingdom, not very far from Leeds and York and Sheffield, that are perhaps a little bit better known than Hull. And really pretty much by coincidence, I got the position, and yeah, I've been here ever since. So working my way up in the UK educational system, you start from lecturer, and then you can apply to be senior lecturer, reader, and finally professor. And that's where I am now in terms of academic career. And I think I leave it there and let Robert take over, and then we can go into some more detail on some of these aspects in the next questions.
Okay, I'll kind of give you a short overview of where I started and how I ended up where I am. Actually, my PhD is in the field of medicinal chemistry from the School of Pharmacy at the State University of New York at Buffalo. And I received my PhD many years ago. It was in actually 1985. And when I was finishing up and I was looking for what the next step in my career would be, it became obvious to me that having a degree in medicinal chemistry, it kind of limited you in what you could do. I mean, the thought would be that we would then, as a natural progression, go work for the pharmaceutical industry. But the pharmaceutical industry doesn't really hire medicinal chemists to do drug development. They've always taken the position of hiring organic chemists because they felt that they needed someone with good synthetic skills, and they could always teach them the pharmacology and biology that they would use in doing drug development. So the options of having a degree in medicinal chemistry were quite limited. And how I got into imaging was quite lucky. I had a friend of mine who finished about a year ahead of me. He was doing a postdoc in nuclear medicine. His name was Simon Ifange, and he was working for Hank Kung at Buffalo. And he started describing nuclear medicine to me, and it looked quite interesting. It looked like a field that was just beginning to grow. And Hank had a postdoc position in his lab, and I joined his group, actually making SPECT probes for technetium-based agents for imaging fatty acid metabolism. A few years after that, I joined the PET program at the University of Pennsylvania, working with Marty Rivich and Rich Ehrenkoffer. And at that time, the timing couldn't have been better for someone with my background to come into the field of PET, because at that time, studies with F18 and carbon-11 N-methylspiperone were just being published. I think the year I joined the field, carbon-11 raclopride, the first studies were being published out of Karolinska. So it became very obvious that there was a need for someone with a medicinal chemistry background, one who could design, was familiar with the drug design process, to apply those skills into probe development. And over the course of my career, and if you look at how my career has evolved over the years and over the decades, I haven't really ventured very far from my medicinal chemistry roots. I try to use medicinal chemistry to develop probes that can be used with PET to study really the molecular basis of disease. So that's how I ended up along my career path. And over the course of my career, I just found myself having one opportunity after another to advance my career, either by going from one place where I was located to another because the opportunities were greater. And you know, the other thing that for words of advice and to assist in anyone who tries to do this line of research, I have been very fortunate over the course of my career to have excellent collaborators, because you can't do science in a vacuum. You can't do chemistry in a vacuum. You really have to understand what the important biological and biomedical questions are. And that's where having very strong collaborators is very helpful. So being able to stick to my roots of medicinal chemistry by identifying good collaborators that could tell me what projects we should focus on, and then being at institutions that provided enormous opportunities for me, enabled me to go from a newly minted PhD from the SUNY system to where I am as an endowed chair at the University of Pennsylvania.
Yeah, thank you very much, Nicole and Robert. It's really great. You know, and the more you explaining, the more question I have. But I will try to stick with the question what we planned. But I just want to remind everybody that our plan is to have 45 minutes with the interview, and then we also have opportunity. And I think we already posted in the chat that people, the attendees, can ask the question. And last 15 minutes, we will have an opportunity to answer some of them. So I have a list of questions through which we will go for the next 30 minutes and see how much we can make of them. And then we will have last 15 minutes for the Q&A from the attendees. And then I will go to my, again, thanks a lot for the answering the first question. And then the second question that I have is, can you identify any specific experience that you had along the way that changed your trajectory and led you down the path?
Yeah.
Nicole, do you want to start again?
You want to start with me again? Yeah, I mean, I think in life, whether it's in life in general or within a career, there are always lots of kind of forks in the roads and trajectories that we go down. And sometimes we think back and think, had so-and-so not told me about LEP on a chip, I would have never known and would have never looked into it, and my life would be totally different. So there are just even individual conversations that I can pinpoint that have led me down paths, or as Robert was saying, you work with collaborators and you just meet somebody at conferences, you start talking about topics, and a few years later, all of a sudden you are an expert in an area that you didn't really know anything a few years before that. But I think generally speaking, yeah, a lot of it it's really a bit coincidence, and maybe some people would call it serendipity or intuitions. I think we kind of make decisions within the framework of what's important for us at a given point in time. So for example, when I moved out of my rural upbringing area into a place to study, I didn't want to be too far away because I had a boyfriend in the village at the time, but I didn't want to be too close for my parents to be just dropping in the afternoon. So I picked a university that was about a 2-hour drive, and there were several places, quite good ones, Münster, Göttingen, Marburg, among those, and then I sort of picked from within those. And for the PhD, I just wanted to be in a big city. So I knew I wanted to do analytical chemistry. I'd sort of specialized in that. But then, yeah, I applied to 3 bigger cities in Germany, and I applied, I think it was Dublin and London. And then I got the London opportunity, which was great. And I took that. And the Japan opportunity, again, was sort of a bit serendipity. My professor at the time had connections there. They were looking for fellows. I had to write my own proposals. I had to come up with an idea and write something. But I wasn't specifically setting out of going to Japan. It was just an opportunity that presented itself, and I took. And very similar with the position in Hull, just a friend told us, oh yeah, there are 3 positions advertised, and he was going to apply, and he suggested to my partner to apply. So they were going to be doing organic and physical chemistry. So I said, oh, I try analytical chemistry, then we can all work together. And I was the only one who got an interview and got the position in the end out of the 3 of us. It was just a bit coincidence, but Hull was very well set up for lab on a chip based research. They had been doing that in particular for chemical synthesis for a number of years, and I was more in the analytical, bioanalytical applications. And at that time, now it's a bit more mature, but at that time it was much less mature. So it was a good time to join there and make a bit of a stamp. And similarly, we've had a lot of investment at the university also in PET facilities, which wasn't something that was decided by me, but it was a great opportunity that presented itself, and where my sort of background in microfluidics played a key role. So yeah, it's just really taking opportunities that have sort of presented themselves at the junctions where I was in life is probably the best way to describe it.
Okay. Yeah, I know that's an interesting question, and I know exactly when a major turning point of my career occurred, and it was when I was at Wake Forest University back in the mid 1990s. And at that time, I was developing probes for imaging the dopamine system, and we were doing some really interesting work looking at non-human primate models of substance abuse and behavioral disorders. And it was good research, and we would develop a probe, we would image. And at that time, as we were developing our probes, we would always screen our compounds for off-target effects at the sigma receptors. The sigma receptors are these proteins that are really poorly understood, but a lot of things would cross-react with that. They're very promiscuous receptors. So if you develop a probe for imaging the D3 receptor, you want to make sure you image the D3 receptor, not the sigma-2 receptor. So I had this assay for screening all my new probes against sigma receptors in a lab. And one day a collaborator came to my office, and he saw this paper where the sigma-2 receptor was identified or reported as being a marker for cancer. And he looked at the data and he thought it could be a proliferation marker. So we started down this path where my lab would be developing probes, and my collaborator's name was Ken Wheeler. He would develop biological experiments to evaluate this protein as a receptor-based marker of proliferation. And it all came together quite nicely. We had a nice probe that was a receptor-based marker of proliferation, and then I developed imaging agents that could image that receptor. And we thought everything was fine and dandy, and we would just start the process of validating it and doing human imaging studies. But the problem in the glitch in it was nobody knew what the sigma-2 receptor was, what it did. And because it was this unknown entity, it really didn't get a lot of respect in the scientific community. So my lab was in a position where if we were really going to advance this science, we're going to have to figure out what this protein is and what it's doing. And at that stage in my career, my lab made the transition from being a chemistry/labeling imaging program to one that had to dive deeply into the field of cell and molecular biology to try to identify the protein, to characterize it, and understand its role in cell biology. So my lab started doing protein labeling, purification, sequencing, CRISPR, overexpression, all the basic tools of molecular biology. And the reason why I bring this up is because it was a turning point in my career, but the techniques that I brought to my lab and that we have learned in characterizing this protein, we now apply to every other protein. Because, you know, the one thing we need to really do, when we develop a probe that images a target, we really need to understand what that target is, what its function is, and what its role is in cell biology. There are some targets where that is the case, it's very well known, and there are other cases where it isn't very well known. And I will tell you, it's personally more rewarding and challenging to take on a project where you have a protein, you're developing a probe where people don't understand what it is, and then you do the hardcore cell and molecular biology to figure out what this protein is, what it is doing, and why imaging it is important. So if I look at the turning point of my career, where I made the transition from being a traditional PET radiochemist / imaging person into one that made that, you know, crossover into hardcore cell and molecular biology. And again, I'm not a hardcore cell and molecular biologist. That's where having good collaborators comes in. That's where that comes in. That's by bringing postdocs into my lab who are you know, trained in the techniques that I was never trained in, to be able to apply those and bring those techniques to my lab, and to really have a diverse approach to probe development. Not only doing the medicinal chemistry to develop the probes, have the imaging infrastructure in place to image the probes, but to really do and use these tools and molecular biology to completely understand what that protein is doing, and how you can design imaging studies to probe what that protein is doing in vivo, in particular in patients.
Great. Thank you very much. Thank you for the great answers. Really, really impressive how you get through the year, and how you get to this, and what you change, what small things can change the path. So the next question I have, so if you could go back in time to the grad school, and what the one things you would do differently?
You want me to start again? So
yes, please.
I can go first. Invest in Google. But or Apple, take your company of choice. But no, it's really, you know, it may be a cliche to say this, but I wouldn't change anything. I mean, I think Nicole pointed out that, you know, in order to advance your career and to achieve any level of success is a combination of you know, having the level of drive, and having the opportunity, and being at the right place at the right time. And if you look at how my career has evolved, I've been the beneficiary of those three things. I like to take on challenging projects. As I said, had great collaborators, and oftentimes I've found myself being at the right place in the right time to take advantage of things to advance my career. So I wouldn't change anything, because I'd probably wind up at a different place, not being quite as happy as I am today. So I'm quite happy with where I am in life.
That's really good to hear. It's not the usual conclusion people give in a sort of a moany coffee break, right? When one sits with colleagues sometimes. Yeah, I have very fond memories from my time in London at Imperial College. So it was very pioneering work at the time. Andreas, my PhD supervisor, is somebody who has so many ideas. You'd sort of go into a meeting and he'd have 10 ideas, and maybe 1 or 2 could be implemented, and you'd kind of had to think a little bit which path you wanted to go down. And we were all doing quite different projects, and so a lot of things I really enjoyed with the kind of way I had my life organized in London. I lived with people in a house share, as you do in London. They were all English. I was the only non-English, so I got a really good introduction into English culture. They were all studying different PhDs in different departments, so we were not all in this sort of same bubbles. And yeah, at the universities in the UK, it's the same, I guess, as in the US. There are lots of clubs, so there's kind of sports and music and lots of different activities and just engaging in things and meeting. I met so many different people, so many different types of people. It's it's really an amazing time in hindsight. The only thing I think in terms of the work I would do differently is maybe be a bit more curious, be a bit more inquiry based as to what the other guys were doing. So in hindsight, I'm saying, we could have done this kind of thing together or could have collaborated there. And at that point, I didn't quite see things like that. We were sort of quite focused on our project and maybe not thinking so much at sort of wider possibilities. So that's maybe the one thing I'd approach slightly differently with my middle-aged brain now.
Thank you very much. It's exciting. It's exciting to see. I think it's a good message for the young scientists, right? We can looking forward to like what we're doing, and then we we will get the good results and get in a good successful career. So my next question is: so do you have any networking tips for graduate students? What is your best networking advice? And
for graduate students, or I mean, I have more postdocs than graduate students in my group, but the best networking advice for them is to, I give them the opportunity to go to meetings, and for them to go to the meetings like the ISRS to present there, to meet people. When you have been in the field as long as I have, and I told you exactly how long I've been in the field, it's over 3 decades, that and when you enter a field in its early stages, like I did, you tend to, over the course of time, get to know everyone. So I think a lot of people, either they know me or they know of me. So being able to go to these meetings and present and give them the opportunity to present is really the best networking thing one can do. The other thing I do, and I think this is important, whenever a postdoc or graduate student starts in my lab, and it's really, as I said, mostly postdocs, I had the same conversation with each and every one of them. And I tell them right from the very beginning, the greatest thing I have to offer you is opportunity to learn. I said, if you look at what is going on in my lab and all of the diverse types of things that are going on in my lab, and I spoke a little bit about it. I talked about the medicinal chemistry. I talked about the cell biology. I talked about the imaging studies, the use of animal models, the translational studies. I probably have 8 different clinical protocols being done with stuff that have been developed in my lab. If you look at the overall big picture of all the research that is ongoing in my lab, you have the opportunity to learn so much. And I tell them, don't limit yourself. Learn everything that you're interested in learning and take advantage of this opportunity. Because if you take advantage of those opportunities and you learn the things that my lab has to offer, then when it comes time to give these scientific presentations, either at the Society of Nuclear Medicine meeting or the ISRS, or I also take my group to the neuroscience meeting. I remember of that. I do have a lot of my research is in neuroscience, so I try to bring members of my group to the neuroscience community. Do good research, learn as much as you can, give good presentations, and you will sell yourself. And that's really the thing I tell all of my students is just that there's opportunity, but you're not going to become successful because of anything I do. You're going to become successful of what you do and how you take advantage of the opportunities that are provided by working in my lab and at a place like Penn.
Okay, yeah, so networking is a sort of a word that we use in a quite general sense. So what, maybe to sort of break it down a bit, I'd start with what is the networking for? What is the purpose of it? So if somebody is a graduate student or a postdoc or maybe even an early career researcher, they might maybe scope out other potential supervisors, other groups. Yeah, so you might want to work with Professor X or Dr. So-and-so or even a company. And how can you network with these people? Just as Robert is saying, you're going to have to go to conference, you're going to have to go to meetings where these people show up. And if you want to meet them somehow, that can be really difficult, right? If you are in a meeting with 5,000 participants, 10,000 participants, even with 1,000 participants, it can be challenging to just bump into somebody. So if you really want to make a mark, yes, as Robert is saying, attend. Ask questions, ideally not obnoxious ones, but asking some questions is always good. Walking the floor to the posters. Yeah, if they are presenting the posters, then you might be lucky of seeing people there. I sometimes am at conferences and people also sort of approach me directly. We've been looking for you, and they introduce themselves. That can sometimes be a sort of a double-edged sword. So if I'm talking to somebody else that's in that moment more important to me, I might end up being sort of slightly annoyed, or I might have really wanted to check out something myself. So check a little bit before approaching somebody if they sort of look approachable at that moment. I've also found that yeah, it's very interesting with different cultures. So in the microfluidics community, we have it's very international. We have many Asian, European, and North American participants at the conferences. And the North Americans will come to me and say, you are the president. I want to be part of this committee. I want to be doing that. So they will really be pushing themselves forward. An Asian person would never dream of that. The supervisor will introduce them, and it would be totally out of their culture. So it depends a little bit on the cultural context as to how you can make yourself known to somebody you might potentially be working with later. But often, as Robert is saying, somebody comes into my lab, people know me in the microfluidics community for sure, or they know of me, and then the introduction I make is oftentimes probably more effective than what the person has done that has tried to do the networking. If you're lucky, both of it has come together, and the new supervisor has seen that student already. But networking is not just only about meeting somebody who one might want to work with. So in this sort of academic careers, people get to know you because you've maybe helped here, been on a panel there, been a chair of a conference, been a member of something. So you sort of start small with that. So even as PhD students, there are often, like we are doing here right now, right? There are sort of events like this for PhD students organized at conferences. Volunteers are sought. Volunteer, get involved. And then if you're doing a good job, somebody remembers you, and then you get involved and asked for the next thing. And then all of a sudden you end up a professor or the president of a society. And because those were things that you have enjoyed doing, and other things you were asked that you didn't enjoy so much, maybe you don't progress. But it's sort of it's sort of one thing often comes to the next if you leave a good impression and have enjoyed doing something. So it's really important going out, being seen. So, and I also see with colleagues that don't travel to conferences very much, they're just really struggling so much more to establish themselves. So, which is why COVID is particularly hard. But with these online conferences, we don't have to travel, and it opens things up as well. So make as much use of them as possible, I'd say.
Yeah, thank you very much. I think that's all great advice for the networking. So, I think we are doing good on the questions. So we have 2 more questions, and then we can go to the Q&A. So the next question would be, what is the most difficult and most rewarding aspect of your current position? So, Nicole, do you want to start?
Okay. So I'm wondering whether to start with the negative or the positive. Let's start with the positive. So I really enjoy research, doing research projects, coming up with ideas, talking with people, writing the projects in collaboration. Pretty much all I do is in collaboration, working with people from different disciplines. I always learn something new. And then you work with people, the graduate students, the postdocs, and you are solving problems together, and they achieve something. And then I don't know, yesterday I was part of somebody's PhD examination, and it's this, it feels really good to be part of, this important days or important times of developments in people's lives. So this working with people in collaboration, achieving things together is something I really like, which is why, by the way, I am not in university administration. I am naturally a good administrator, but ethics committee meetings and strategic meetings for hours and hours, and 3 months later you are still discussing the same stuff you've already discussed 3 months earlier, and probably 10 months earlier than that as well. So that wasn't for me. So this is the kind of thing that suits me quite well. I think the most challenging is work-life balance, to be honest. When you were asking, what was good in graduate school, I did that much better in graduate school. I went out and had a lot more hobbies and had a lot more of a dance. And this is much more challenging, because the kind of general attitude in academia is often work more, work hard. If you work hard, you get somewhere, and you kind of take on and say yes and take on. And eventually you come to some kind of breaking point, and sometimes people can sort of gauge that, and you have to learn to say no. But I think, and sort of in my life, we have 2 children. I had them relatively late, in my mid-30s, and that really puts a spanner in the works. You've sort of worked yourself up to a kind of a certain plateau of activity, and all of a sudden there's something there that takes a lot of time, and then you realize, you can't deliver exactly on the same level. And even during COVID times, there's so much extra stuff that is coming our way. So this balancing it and taking the time out and not working every evening and not working every day of the weekend is something that's a sort of a constant struggle, I'd say.
Okay. Yeah, for me, now being an academic scientist, the most challenging thing I face, and the one that I struggle the most with, and I think everyone in a similar position struggles with, is getting the extramural funding to support your research. It really is a big challenge. It's something that is necessary to do in doing biomedical research. And I will tell you that most of my time in getting the grant funding to support my group goes into the process of submitting a grant, the strategy in submitting it, how it is presented. It is focused more on the grant submission process and how the data and how the science is presented, more so than the science. And that's something that you have to learn over many, many years as to how to do that, and how to write grants that are easy to review from the side of the reviewer, because they're in a very difficult position. They have a bunch of grants to review, and they have to identify the good science and the projects that are worthy of being supported. So they're in a very difficult position, and learning the tricks to the best way to present your science so that you get yourself in the position of being funded is a real challenge. And it took years for me to learn that process. And going back to the previous question, I think that's where the networking is very important, because going out and being involved in the either national or international societies, there are opportunities for people in the early stages of their career to get involved in these societies. So taking part in that and getting to know people is extremely important, because there's a very good chance in the future that one of those people who you interact with may be on a grant review committee, and having that name and face recognition is one of those intangible things that ultimately will help you. Making good scientific presentations, and so that you remember you, and networking like Nicole just said, and going up and talking to people and not being shy and introducing yourself is another way of doing that. So the most challenging thing that I've encountered in my career, it is a constant struggle that I sit there. It's like, okay, I have a project, I have really good data. Now, how am I going to get this thing funded? What strategy do I have to use? And there's a whole science behind that. And I could spend a whole hour talking about how you do that. So I won't do that. But it's the most difficult part of my career, is maintaining the funding to support the research coming out of my lab. The most rewarding thing is when we take a tracer all the way from, you know, a structure on the whiteboard in my office through the entire validation process, and then we do our first in human studies. And I always make it a point that when we do our very first in human studies, first in human study, I am at the scanner watching it being done. And the reason why I do that is the amount of satisfaction you get when you see that patient receiving a tracer that you develop, and then you watch the study being done, and then when the data comes in, and if it even works, that's better. I mean, it's just it's rewarding to the point of being addictive. Right? So it's really the most satisfying part of my career, is to take something that is an idea, a structure that is an idea, and then seeing it mature all the way to a clinical study that is producing hopefully very important clinical data. Okay.
Yeah, thanks. So that's it's really, you know, it's really great. I'm really pretty much echo, you know, from every every single word you both Nicole and Robert said about the challenges and rewards. And you know, and the last question I have for the, you know, the list of questions we prepared, it's what message would you want to send to the next generation of radiopharmaceutical scientists?
Yeah, maybe just almost picking up a little bit from what Robert was saying there, we tend to have a kind of an idea as to what a professor does and what a professor is. And sometimes when I go to such career events, or also sort of women in science events, I tend to give a little bit more of an overview of what a career really entails. And just as Robert is saying there, we spend so much time writing grant applications. It really takes a lot of time. And then you get the grant finally, and then you have to find a person to put your vision into reality, because it's not me standing in the lab. I have to then instill this this vision into a person and lead people. And so these were all things I was really naive about before I went into academia. You know, I was good subject wise, and I enjoy learning. I was, you know, good at the bench. I could do the stuff. I can give a presentation, but this the constant dealing with this being rejected, and like this bouncy doll having to sort of come up again, and your first rejections, it takes you a lot longer. Then you get a bit more used to it, your skin gets a bit thicker. But if you've really put your heart and soul into it, oh, it's hard, especially if it then comes like on a Saturday. Some funders tell you on a Saturday evening, you know, really. And yeah, the people management, this was something I was really, I mean, I did a lot by intuition and learned, and then later in my career, I've been on leadership courses and have understood so much more about myself and about leading others, and intuitively I'd sort of applied a lot of things like that. So having a bit of an awareness that that the career is not just about the science we are doing, but so much more around the soft skills is maybe one message I'd give. And I guess for radiochemistry in particular, so, you know, somebody might think, oh, you know, radiochemistry, who needs that? It's been around for a while. Scanners have been around for a while, but so much is happening, not only on the biological and chemical side, where sort of Robert has been explaining, but also with the tools, which is more my area, the microfluidic tools, the mini cyclotrons, that will open up radiochemistry to so many more people, and hopefully then allow us to develop so many more tracers and try so many more things. So I think it's an exciting time to be working in the radiochemistry field.
Yeah, I completely agree with it. I think this is a very exciting time, and I think radiochemistry and imaging and molecular imaging is going to have a key role in the advancement of science, particularly biomedical research in the years to come. My advice to to the young people out there is it's kind of following what Nicole just said. Is number one, don't be afraid and don't take it personally if you fail. I mean, for every success that I've had in my career, I've had 10 failures. And I've had some very antagonistic people out there who told me exactly why I was a failure. But you can't take it personally. You have to develop the thick skin that Nicole mentioned and use that and channel that frustration. And in my case, I think it's pretty bad when it happened, but channel that in an effective way to overcome it so that you learn from that failure so that the number of failures decrease going forward and you start to have some success. But in science and in academia and in this type of research, you are going to experience failure. Don't let the failure, don't be afraid to fail. And you have to know how to deal with failure and overcome it in order to become successful. And number two, don't place any artificial barriers around you. You know, science is science, and you need to go and follow the directions that science takes you. And oftentimes people like try to put these barriers up. And I'll use an example. A colleague of mine that collaborating with, I tried to explain one of the things I was working on is a very, very famous person in neuroscience research. And they looked at it and they looked at what I was proposing. I said, well, that's a tumor biomarker. That's involved in cancer. It's not involved in neuroscience. Well, it's you know, we know from work that we've done that that's not the case. And really, that's not how biology works. Biology, these are proteins, these are pathways, these are biological mechanisms, and how a protein functions is dependent on the tissue. It's tissue dependent and context dependent. So don't say, oh, I'm going to study neuroscience, or I'm going to study oncology, or I like cardiovascular research, right? If you do that, you're kind of narrowing the opportunities that can be presented to you. And I've never put those barriers up. I've always gone to where the science has taken me. If you look at my publication list, you see I've published in neuroscience, in oncology, in cardiovascular disease, and lung injury and infection. I've always I haven't been hesitant, and I don't put barriers on myself in going into new areas, because, and how I explain it is, what I do is essentially the same. I'm a chemist, right? What I do doesn't change. I'm a chemist, and I develop probe, and I do radiochemistry. The science that I do depends on, the concept I mentioned earlier, the collaborators I work with, and the biological questions we go after, which are largely driven by the collaborators I work with. So my advice is, go where the science takes you, and don't put any artificial barriers up that keep you in one area of science and afraid to branch off into a different area. Because my whole career has been able to pivot and go from one direction to another. And the reason why it's important is going back to my prior answers. Like, there are times when you're working on something and the field it's not particularly hot right now. So if you're working on a project like dopamine receptors and it's not hot right now, and you still want to work in that area, but it's not popular, you need to pivot and do something else that is popular to maintain the funding in your lab. So being able to pivot from one area to another to maintain a research lab is critical. And it's a lot easier to pivot from one direction to another if there are no barriers in place that prevent you from doing that. And I've had to do that time and time and time again, because I understand that what isn't hot today is very likely to be important tomorrow and will come back. And what is hot today is not likely going to be important a few years down the road. So you have to be able to see the trends as they occur and be able to make that pivot and be flexible to be able to adapt the research in your lab so that you're able to maintain the level of funding one needs to do research. Things will always, science is like everything else, it cycles. Projects that are interesting today fall from favor, they will come back in the future. So being able to make that scientific pivot from one area to the other is extremely important. So don't be afraid of failure, and don't place up artificial barriers that keep you from going into different avenues of research.
Great. I think it's a great advice, really great. So I think it's will helpful for new scientists and young researchers to lead them to the successful careers. So we have now I think we should go to our Q&A session, and Ima will lead this session. So I saw a couple of questions already in the chat. So Ima, do you want to take it from there? And thanks again, Nicole and Robert for being on time and for the great answers.
Yeah, thank you very much. It was great to hear your stories, and I think just kind of a take-home message to like take all the challenges and like new projects that come to us, and just jump into them and like learn from all the collaborations and everything. So we had a couple of questions. The first one saying, as a medicinal chemistry PhD student, is it necessary to have very advanced knowledge in organic synthesis? I don't know if Robert or Nicole, any of you.
Yeah, it's probably more direct.
Probably Robert to go first.
Yeah, I think, having a strong understanding of the organic synthesis is important, because again, that is a tool that you are going to use. So I would recommend trying to hone your synthetic skills, because if, as you design molecules in the future, you either need to be able to do it yourself at the early stages of your career, or you're going to have a postdoc or a student who will be struggling in the lab, and you have to give them advice as to how to overcome and complete the synthesis of the project. So yes, I think as a medicinal chemist, having a strong synthetic background is important, because that provides the foundation for all the probe development you do.
If you want to keep working in that area, right? So if you hate organic synthesis, then maybe think of other opportunities where you can apply medicinal chemistry, sure. but don't do the synthesis. Yeah, the sort of supervising something where I'm not quite an expert challenge is certainly a situation I also recognize. And just as Robert is saying, if I'm supervising somebody who's doing computer simulations, I'm sort of struggling at the first hurdle they have. So then you have to bring in collaborators or other expertise. I hope that your researchers or postdocs are good at that by themselves. But yeah, if you really hate it, and then within medicinal chemistry, there will be lots of different paths.
Sure, absolutely.
All right, thank you. And then the second one says, when you're developing a new radiopharmaceutical, there are a lot of steps to perform prior to the clinical. From your point of view, what are the points of the CMC that are the most challenging?
Yeah, that would be for Robert again.
Okay. For the CMC that is the most challenging, well, I think, well, I don't know how challenging they are. I guess the challenges become less and less the more often you do it, but clearly being able to have an optimized synthesis so that you have a very high success rate when it comes to making the tracer for a clinical study. That goes back into the design of the probe. And if you look at my probes, and especially the ones that have made it into the clinical trials that we're doing, the chemistry is really not that elegant. I mean, I think that the elegance comes in the design of the molecules, not in the radiochemistry we do. And so I try to simplify the radiochemistry, because I know it's important as part of the CMC to have a synthesis that is very streamlined, that gives a very high quality, specific activity, radiochemical purity. So you want to have a simple synthesis that can be adapted to the chemistry modules that we use to simplify the challenges in putting together a CMC section. Having the toxicity data is always a challenge. You need to be in a supportive environment to get the resources you need to do the toxicity studies. But the other thing is you have to have. It's helpful to have an infrastructure like I've had previously at Washington University and currently at the University of Pennsylvania, who are experts in actually doing that. So I don't really get involved in the in doing the CMCs. I have a trained staff that are very good at doing that. It's my job to make their job easier by simplifying the radiochemistry so that that the CMC section is easy for them to write. So that comes in the design of the molecule up front.
Great, thank you. And I think there are no more questions in the Q&A, but maybe I'll just like to ask to add one more question, and then maybe Felice and Valerie want to follow up. Did you ever had the industry, and if so, like, what I want to stay in research, or I don't know, like, I think for grad students it's like, yes, we love research, but do we want to take up that challenge to the end, or do we want to switch? Like, have you tested the industry and then move back to academia? Just I don't know.
Yeah,
Go
I
ahead.
guess I'll go first. And you know, I will tell you that in recent years, and I'm sure this is something that everyone, all the young people are struggling with on this call, because my postdocs and graduate students are going through the same thing. And we've actually talked about this at the graduate program here at Penn, is where the number of trainees who do not want to go into academia, but want to go into industry is very, very high. And I think what they see is, you know, the challenges that I mentioned earlier in maintaining a research lab and getting funding. And a lot of those people just don't want to go that route. They want to go into industry. But industry, I've never had the opportunity to go into industry because I never wanted to go into industry. Because, you know, no matter what you do, you're going to have challenges. You're never going, unless you're independently wealthy and did invest in Google, or your parents invested in Google big time 20 years ago, then whatever you do, you're going to face challenges, and you have to be comfortable in accepting the types of challenges you face. In industry, it's a stable environment. You don't have to write grants, but you're always faced with a decision of, well, we're closing down this section and we're laying off 1,000 people. Now you have to find another job, right? And that is a scary scenario for me. I would much rather sit in my office and scratch my head to figure out how I can get this grant funded, and pursue that challenge, than is be at the mercy of decision of an MBA in industry who decides we no longer want to do cardiovascular research, right, or neuroscience research. So you have to really pick your challenges, and the reason why I describe that is that it is very real. But the point I want to make is, it's like everything else, it's a learned process. You just need to understand what that process is. And if you have good ideas, you will eventually be funded, but you can't be afraid to take on these challenges. And I'm very happy in academia. I can find it frustrating at times, but as I said, if you're able to accept and deal with the failures and know how to overcome those failures and keep working at it, you will eventually be successful. And the thing about academia is that you are in the control of your destiny, right? In industry, I see other people being in control of my destiny. And I would much rather be in control of my destiny and be responsible for my success or failure than have some MBA, Harvard MBA come in and decide that I'm no longer needed, go find a new job. So it's life is full of challenge. You just have to be able to accept a challenge that you're more comfortable with, and then make decisions accordingly.
There was a great endowment for academia. Yeah, it's and that's the typical answer you will get from any professor, right? We're sort of here by by choice usually, because it tends to be, finding a job in industry is often easier compared to getting an academic position. They're sort of they tend to be very competitive, and there's no way everybody that's studying could end up in academia, and often people are on this sort of one postdoc after another, and eventually they do this kind of jump into industry. So I've worked in industry short periods of time as a summer student, earning money on production line, packing plastic bags or drilling threads into metal pieces for 8 hours on end. So I remember vividly being shocked that there was a mother and daughter in the coffee room, and the entire coffee break was complaining about the job, yet the mother's daughter was sitting there next to her, and I just could not understand that. And Yeah, then I did work while studying in a couple of chemical companies, one in Frankfurt on the Höchst campus, a sort of polymer company, and then also near London for ICI Paints at the time. And yeah, I mean, I don't know, the vibe in these places was just very different. You sort of come in as a young person, often new to the area, and there are all people that are sort of middle-aged or older that are getting on with their lives and have their own stuff going on. So it feels very different from the sort of vibrant university environments that we are usually embedded in. And yeah, you know, it depends how creative you want to be, or to what extent you want to do what somebody tells you to do. So a lot of our students work in industry, and they do a year in industry. I go on visits. We sometimes have career events where people talk about their career in industry, and some of that sounds quite fascinating. And there are people that do really cool projects, setting up sort of, you know, big things and leading things. And at the other extreme, you have sort of somebody coming in just as a measurement scientist, but you can rise and you can go different career paths. You can go into the patent side, practical side, legal side, marketing side. There are loads of opportunities in industry, but ultimately the point is to sell a product. Everything is just very product oriented, and just somebody might just decide they are closing things down. So, for example, Philips in Holland, they did a lot of microfluidics, lab-on-a-chip diagnostics, lots of it. And there's somewhere there in a small town, half the town there works in diagnostics, and somebody decides to shut it down. And you're settled there with family. There isn't going to be another job in diagnostics for you there. So you sort of have to be ready for that risk. So either you then change and do something else, or you have to move. In academia, it's rarer that university departments get shut down and people are sacked in academia. It happens, but it's much rarer, and you have the creativity, where, so in Robert's case, he finds it very rewarding when the tracer goes into the patient. And yeah, for me, we are sort of doing analytical measurement instrumentation. So if somebody uses this ultimately, or if it works, and you've collaborated with, yeah, I don't know, it can be people on the clinical side in European countries as well as in lower and middle income countries, but it's rewarding. It's your idea. It's got your heart and soul in it. In industry, you don't tend to have that so much. You're part of a bigger team, and it's often you're sort of implementing somebody else's idea. But the salaries are higher. There is a steeper trajectory, but there's also a riskier environment. And I know my personality, and I thrive under security. I'm not a risk taker naturally. So I also, like Robert, like to sit in my office scratching my head rather than having this sort of insecurity hanging over me. But some people thrive in that. They absolutely love that. So it's a personality thing.
All right, great. Thank you both very much. I think we have one more question, but we're 6 minutes past the time. So I believe we won't have time for this, but we encourage everyone to just meet Robert and Nicole during the ESRS. Tables and the setup we have in Remo is just going to be great to like drop in a table and ask as many questions as you guys want if they're there, and just chat and network as much as you can during the ESRS. So if Melissa and Valerie, do you have anything else you would like to add?
I wanted to thank our speakers again. This was a really lovely session. It was rare that you get such an honest and thorough examination of academia and still leave feeling optimistic about it. So I really appreciate that personally, to hear a lot of the challenges that I go through, but still excitement about what you're doing, and the idea of how, you know, you still get moments of inspiration and things that continue to keep rewarding you to keep going. I appreciate. I also wanted to mention really quickly to the person whose question we didn't get to, there is a debate during ESRS on that very topic, so there will be a lot of discussion of that to come. But otherwise, Valerie, if you have anything else to say, just thank
you. The same as me, because I just want to thank you a lot for the very nice interview. And I think, the goal of this interview was to connect the young scientists and young researcher that they can, come and ask their questions also during the ICRS. So I think I think we achieved it because we have more questions than we have a time, which I think it's a good sign that this question can be asked during the meeting next week. So really thank you. I think it was a very successful meeting.
And also thank you to Charles and Bailey for helping us set all of this up, because that was very important. All right.
Okay.
Great, thanks for having us.
Wonderful.
Yes, thank you.
Thanks everyone for coming, and hopefully we'll see a lot of you again next week.